Hello. I want to repeat our last speaker and first say thank you to Medicinal Genomics, Advanced Nutrients for the invitation, and to probably address the question that you either saw reading the agenda, or you thought of sitting here, or you're seeing with this title. What in the world can a plant scientist from Texas, a state with one of the most challenging, deficient, or add your own expletive term, medical policies have in a conference full of medical practitioners? And I would hope, with a little bit of transfer of knowledge and exercise, I can demonstrate that it is. And the first request I have, everyone that's here, if you have a cell phone, would you hold up your cell phone? Does anybody here not have a cell phone? All right, every single one of you is holding a handheld phenomic tool. You're just playing games on it and not using it for what it could be used for. That's my transfer of learning exercise. All right, my background is perennial grasses, 16 plus years, 3 in cannabis, so I'm not a legacy breeder. I'm an upstart, a noob. I have a love of viewing plants as biorefineries, integrated biorefineries, and this is from brilliant scientists in Germany who corrupted me, influenced me back in 2006 and 7 when I was turning grass into ethanol or biofuels. And I was able to put on a second hat, which is good for me. I am follically challenged. I not only work with perennial grasses, I now work with industrial hemp. And industrial hemp, to me, is not one commodity. It's as many commodities as you can create from that plant. It is an integrated biorefinery. It's built that way. It may be the best example of a plant biorefinery out there. Okay? If I leave out all the perennial grass work and just focus on what we're trying to do in hemp, it is a biorefinery, and I'm going to boil this down even further to our handheld phenomics scope for this talk. But our breeding program views it as such. We're working on multi-use hemp, fiber, grain, cannabinoid, any use of those, anything you can convert with it. And as a plant breeder, hemp has not been advanced in a conventional sense compared to corn, other major crops in 80 years. So a lot of our work is to make modern types of cultivars, pure lines, F1 hybrids. Put your plant patents on one, your plant variety patents on the other, and you have rock solid intellectual property. Okay? Tetraploids, things I've done in perennial grasses, it's popular in hemp now. Triploid, seed steril, these are your seedless bananas, seedless grapes. You do that with CBD hemp now, there's no concern for pollen. Type 5s, everything in this conference has to do with type 1s and 2s, a little bit of type 3s. Type 5s have their place. Texas, southern US, we're here in Florida. Heat and drought tolerance, if we're going to move this crop outdoors, as some have proposed is important, that's part of our work. And when I started working in hemp 3 years ago, I had absolutely zero seed in my seed room. So trying to develop a germplasm collection that's available, it's public, and it's adapted to subtropical and tropical latitudes is also a big part of our program. And if you look at the very bottom of that picture on the right, that's really where we are today. I'm going to try to introduce just 3 little components to try to make my job easier as a plant breeder, where we need things to look at plants, get data points rapidly and cost efficiently. And I would say there's not an argument of university versus industry doing this kind of research. We are university scientists. We are an arm of industry for their research. Okay? That said, disclosure, I do work for a behemoth of a bureaucracy, universities, and we are regulated by the states, as are in all states that have cannabis policies. That said, we have a lot of supporting offices at A&M that are involved with hemp. We have a beautiful office that helps us engage with industry. We have faculty and extension that are running trials on cropping systems and how to get this outside. We have people working on hop latent pathologists. We have gene editing being conducted. And we also have students very, very interested in this, so much so they have started the only university organized club around cannabis and hemp. Okay? That said, repeating to some of the ethos of this conference, I'm going to propose broad spectrum, if not full spectrum, but broad spectrum approaches of what we all do, what you all do. You're bringing light to cannabis. Okay, and these phenomics tools are around 3 specific portions of that spectrum. The first tool, this has been the toughest for me. I've had more failures than I would like, but not as many as Edison had, and we have a tool now that's very interesting, and it's based around near-infrared. Near-infrared has been around for decades. In forages, we use that to quantify all sorts of composition and quality. So it's not a stretch to say that's already in the book and canned. Miniaturized versions of these, they were put on cell phones 7, 8 years ago, 2016. It would just make your iPhone 3 times as expensive. Not much of a customer base for that. But miniature NIRS platforms are there, they exist. Workflows that are very typical of these types of approaches: screen large populations, sample, develop your modeling, validate those models, put it to work, commercialize it. There are already 2 miniaturized NIRS platforms specifically for cannabis. You all, I'm sure, aware of these, the Jessup and the Purple Pro. These are both based on diffuse reflectance. They both require dried, cured samples. They do give you a cost savings versus HPLC or GC. One of the biggest reasons they have not been adopted widely is you cannot get really high precisions or accuracies, 2 to 4% in most cases. And they may argue you can do this in real time, but in reality you have to have a dried, cured sample. They have to avoid the water bands and those reflectants. So it limits you in those 2 respects. And that's kind of the problem we wanted to tackle with this as a breeding tool. In the last 3 years, thankfully, we've also been able to develop our germplasm, and we have large segregating populations for THC, CBD, and many of these cannabinoids. So we have the NIR, we have the populations, and I've done phenomics in my lab. We've done these things with UAV data. We've done this with ground penetrating radar. So the analytics side and the modeling is there. What's missing, and we wanted to tackle with this tool, was the sampling. What and how you're going to sample, what and how you're going to take the data point, and how you're going to process that. And I apologize for this being a black box. This took longer than I wanted to take. We do have 3 very novel, very non-obvious components of how we're collecting this data, how we're sampling these plants, and we have enabled this to the point where it is accurate much more than the 2 products on the market. And in addition, it lets us sample dry or wet tissue. So it can be very non-destructive. It can be done anywhere in the life cycle of these plants. And that gives us something that is patentable. We've disclosed that. And that bureaucracy I mentioned that moves slow, sometimes slower than rosin coming out of a freeze dryer, is still reviewing it. So the plan is to put IP protection on these, and that license will be held by our sponsors who are here today. Rare Earth Genomics, they enable this work. Again, we are the research arm in this effort. And if you'd like to visit with them, if you haven't seen them yet, please search them out or come find me and I'm happy to introduce you. All right? Moving from infrared to visible. This has been something as an academic, professor, undergraduate classes, graduate students, the student clubs, students in my lab working on trichomes. These beautiful structures, silicon dioxide, full of things that we use, and the colors that they reflect. Trying to gauge trichome size, maturity, and their other secondary phytochemicals. This has been very easy to adopt with the advance of your cell phones and very, very small microscopes you can put on those phone attachments now that have beautiful optics, almost to the stage of 1000x ocular magnification. You combine those with those 2 apps, almost any person within half an hour can get pictures of trichomes at 600x magnification, calibrate those scales, and get your diameters. The larger trick is to try to quantify how much you have on a bud, on a bract leaf, in a sample. You can do that with a little bit of image analysis. These are older technologies, they're 20 years old. ImageJ, OpenSea, you can use these in open source scripts. In the end, you're making grayscale images and looking for circles. The trichome size is one of the easier ones to attack with digital microscopy with your cell phones and a little bit of image analysis. Maturity is much more difficult simply due to the fact that when you use these algorithms, they're all based around grayscale images, so that you lose all of that data you need to gauge maturity. So we struggled with that. We looked at different approaches. We looked at different things we can pull out of those images. And recently, it dawned on me to stop thinking linearly, stop thinking in 2 dimensions, start thinking in 3 dimensions. Every image you take is not a 2-dimensional image. There's data in those pixels beyond RGB. If you look at the additional data, you can project that data and make a 3D rendition of that. So any computer science kid that uses these graphics to make mesh video games or cartoons or comics or videos can do this. You're projecting some portion of the data in that 2D image to get more 3D projections of it. And the first thing we noticed was an immature trichome is clear, optically clear, there's nothing in it, so all you get back is the green of the leaf. The first difficulty in doing that is to get rid of the green. In today's world, we have blockbuster films that are covered in green screens. It's very easy to remove the green. And in our case, we found not a need to remove all of the green, but some of those green spectrum. With that, you're left with a lot of trichome data and what's in the trichome. And if you will take that, minus the green in the middle row there, across maturity of trichomes, you can then pick, you can experiment with what versions of data you would like to project. And what we found is looking at luminosity or the overall brightness of those pixels. You can project those up vertically. And if you work with the saturation is simply the pure colors of those pixels. We all like highly saturated pictures. We like pure blue, pure red. And if you do that, you end up with 3D projections. You can see on the left, immature, largely blue pixels are projected up. Fairly mature trichomes, largely green, some yellow. Very mature, sometimes overly mature, lots of orange and reds. And the beautiful thing is once you have these 3D projections, if you go one step further and quantify the degree of redness, you could calibrate this to harvest at your optimum degree of amber trichomes, depending on what your target is. Okay? Trying to streamline this for breeders and other practitioners, if you can envision doing this at a lower magnification, you would make it easier for someone walking through there with a cell phone, which can almost get you to 20x optical magnification, and produce images like this of an entire bud. You follow the same workflow, remove the green, do a 3D projection, you can get an absolute estimation of the maturity of the trichome composite on those buds. Right. Last tool, this is the UV. This is something going back to my undergraduate and graduate school days. Doubling chromosomes in plants is much easier than animals, almost impossible in animals. In plants, you get double dosage of all those genes. You end up with larger cells. And in hemp, that gives you two things of great value. It gives you larger seed and longer fiber. I've done this in the perennial grasses. We did it with the cannabis. The biggest headache with polyploids, the chemical you use, or colchicine, inhibits mitosis or spindle formation. You end up with part of the meristem being doubled and part not. So you end up with chimeric or mixoploid Frankenstein plants. So trying to determine if you truly have a tetraploid plant is the bottleneck. This technology has not improved in my career. You can either count chromosomes, which is very laborious, or you can use a flow cytometer, which is simply a capillary system using a laser to reflect stained intercalated nuclei. And with hemp being a resinous plant, you can imagine what that resin does to the capillary tubing in those systems. So it's fraught with errors and it's extremely time-consuming. Our approach has been to develop a microfluidic system. You know, Agilent was next door in the expo hall. A lot of our tools these days are becoming miniaturized by simply going from capillary systems to microfluidics. No one's done that yet with flow cytometers. You can do that yourself. We're working with it with fluidic cells you buy off the market or those you make yourself with 3D printers and some imagination. Same isolation of nuclei, microfluidic separation. With or without electricity, it works either way if you want to do pressure. And staining with your fluorochrome of choice. We use ethylene bromide because it's cheap. And in that case, it puts you in the UV range for illumination. That said, this involves breadboards, this involves coding, this involves a little bit of ingenuity that my students didn't have time for. We were faced with biological deadlines and deliverable deadlines for contracts. We had hundreds of plants we wanted to screen for polyploidy. So we went back and used tools that had been done previously looking at cell size. Part of the cell wall, or stomata. Diameter of that has been related to polyploidy for decades, and the advantage we have today, we're much more precise with image classification and with digital quantification of this versus our own visual eyes. Doing that, looking at stomata off of lifts of epidermal cells, and developing our models between diploids and tetraploids. We've gotten very good at being able to predict if these are going to be diploids or tetraploids. And this allowed a student to look at 800 plants in a couple of weeks versus doing it with a flow cytometer and taking a couple of months, knocking on wood that everything works and nothing breaks. So either one of these systems, the microfluidic version or just the imaging version, could be great tools for people that are in the business of looking at polyploid in cannabis. All of these products have been funded and enabled by our research arm, Rare Genomics. Their CEO is right there, their founders are there and up here. I encourage you to visit with them. Without them, my hemp program would be a lot smaller. I would still be much more of a perennial grass breeder. And they have other tools as well. If I go back, they've enabled other work as well. We've developed things like terpene-based insecticides that are going to be disclosed very soon. We've developed a huge germplasm collection. They now have a suite of tetraploids, of inbred lines, of synthetic hybrids in bulk populations. So they have a huge hemp germplasm collection. And the next year, we anticipate disclosures on nanoparticles, nanocellulose, and biocomposites. So it's going to be a pretty good deck they're going to have for IP around hemp and cannabis. That said, I teach a class in hemp and cannabis. Our department head, who is also here, teaches a class in hemp and cannabis, and we would love to teach more classes. If anyone here has interest in trying to help us develop a medical cannabis curriculum or any other one that might be of interest to you, I would love to visit and see if we can build it into some sort of a combined class. And this can be undergraduate, graduate, online, CEU, whatever you can envision. The student club I mentioned is here in Forest. Its founder and president, Clay Moore, is here. You might have seen him around or visited with him. All overachievers. If you want to have student involvement, please reach out to Clay. And again, just turning back to the narrow focus I tried to get to with these three tools that relate to medical cannabis. That link is to a shared drive with the one-pages of almost everything my program does, most of the projects. So if you'd like to visit with other potential topics around this biorefinery plant, please feel free to reach out. And I know Ben's not up because I always finish early. Questions are welcome.
Were you saying that you were unsuccessful with the flow cytometry to identify ploidy? Was that a challenge to do that?
The conventional flow cytometry?
Yes.
There are several problems. One, the instruments are old. And two, the capillary tubing tends to get plugged up by the cannabis resin, and it encourages mold growth. So you're always trying to clean out and disinfect. You spend more time disinfecting that tubing in that system than actually running your samples.
So it's just an inefficient process.
Inefficient, and the machine still costs 30 grand. I mean,
Okay.
it's crazy expensive. You can take a digital image of a trichome lift using super glue and a microscope slide of the epidermis and classify that stomata, get the volume, and as long as you have your comparison of diploids and tetraploids, you'll know which class it's going to fall into. So it could be a very simple digital imaging solution versus that old clunky flow cytometer.
Okay. Yeah, I've seen the flow cytometry used. Just, I didn't know if you were successful or if it's an efficiency
works.
thing. Yeah, okay.
It's torture for students.
Sure. Well, we're all students too, so.
Sure.
I thought the ability to quantify the degree of amber hue of trichomes was a really interesting development, and would go a lot further than me having a Zoom call with one of our growers saying well, how amber was it? What was that eyeball percentage? So I'm curious if you found any correlation between that and concentration of CBD, dry matter development, anything like that?
They're separate tools. We haven't tried to do any combined analyses. That's a beautiful question. We're much further along with the NIR. We can use that very easily to quantify. The 3D animation, it comes and goes with student interest, and the transition to trying to make the 3D mesh is fairly recent. So trying to relate that back to the actual composition of the trichomes, we haven't done it yet, but you would fully anticipate it would be, as you would think.
Cool. Thank you.
All right. Thank you.